currently working as the assistant professor at Rajganj University, Uttar Dinajpur. He has obtained his PhD from Indian Institute of Technology, IIT Kharagpur, India. He has been trained as postdoctoral fellow at the New Jersey Medical School and University of Washington at Seattle, USA, on different aspects of HCV, HIV, and AIDS. Coming back to India, he has been associated with National Center for Cell Science, NICCS, Pune, as research scientist. Before joining at the Rajganj University, he served as research assistant professor at SRM. Some of his achievements or some of his contribution to this field are assistant professor at SRM University, Tamil Nadu, India, for more than 3 years. He has obtained his PhD from Indian Institute of Technology, IIT Kharagpur, India. He has been trained as postdoctoral fellow at the New Jersey Medical University, as I have stated before, and he has came back and he has had many different contribution to this field as well as being a very highly educated person. It is without any doubt that we are very excited to have him among us and we cannot wait to hear some of his words. Sir, please take on the stage.
Okay. First of all, thank you and good morning to all of you. Am I audible to all of you? The voice is okay?
Yes, sir. Yes, sir, you're audible.
Okay, so please feel free to stop me if I am not, audible or if you have any doubt in between. Okay. And thank you. I don't know what is your name for your, kind introduction for me. So what I have understood is that it's a forum where mostly people are in the undergraduate level, right? Am I right? So you are in the undergraduate level, right?
Yes,
Hello.
sir.
Yeah.
Yes, sir.
Yeah. So that's why I deliberately make my talk such a way that I will not go vertically. Instead, I will just show you or ignite you whatever you learn in your notebook day-to-day basis, syllabus-wise. So how that is really applied in a very serious way to tackle, a real-life situation, real-life problem. So I have chosen two. So I'll be talking about modern diagnostic approaches. Because of this COVID pandemic and several other, pandemic and related stuff which has come out recently, we have understood that diagnosis, like especially in case of India, where we are still developing country and there are lack of resources. It is not evenly distributed to very low resource setting area, rural area and all. So the use or the screening of diagnostic has been, understood uh, quite well because of this pandemic and all. So as I am working on some of this field, so I have jotted down two aspects, and I want to present before you guys, so that you can ignite and you can understand how you can utilize whatever you have learned in your textbook, in your curriculum, to tackle some of the problem. Okay, so I will be dealing with two things. One is it's a dual detection system for dengue viral infection. So as you know, dengue virus is a tropical disease, and we and some other countries in the subtropical region, they are quietly widely affected. So coming back to India, I wanted to contribute as I was a trained virologist, so I just devote myself to find a portable point of care devices which can be utilized in a very rural setup where there are not much resources available. Okay, and the second one, which I yesterday night only I jotted down. I was thinking what to add. So I found recently that. Uh, that very bad shocking news who loves music that KK has died, like the great musician KK has died, and this is because of cardiac arrest. So far what we have understood from the newspaper, from the social media and all. So as I am also trying to one project is going on, and that is based on cardiac prognostic kit. So I will be putting before you some of the aspect of what we are thinking and what the future aspects are. Okay, so with this little background to what I'm going to do today, let us start with the first one. It's like liposome based dual detection system for dengue viral infection. Okay, now to quickly take you to the dengue, I think all of you is exposed to this disease. Some of your relatives, some of your, because we the Indians or the subtropical country people are very much aware of the dengue virus, dengue viral disease, even dengue hemorrhagic shock syndrome. So dengue, as you know, this is a vector-borne disease. Mosquito is the carrier. It's a tropical disease, and the latest statistics shows that around 50 to 100 million people yearly got affected, and around 5 lakh cases of hospitalization, and more than 20,000 people annually died because of this disease. And add to that some of the comorbid condition associated with dengue, like if you know in a very so what we know, like Indian, every rainy season we have dengue fever, Japanese encephalitis, there are so many viral fever, but we didn't pay much attention until and unless we get some pandemic and some epidemic kind of thing. So where a lot of people, especially children, got died. So you know the in the later stage, like when it is not no longer confined to just a dengue fever, there is a condition comes called the dengue hemorrhagic fever. Okay, that in that condition, you know, this is a very bizarre situation where you know people get blood oozing out from their nose because your platelet count has drastically gone down. So this is a condition. It's called the irreversible situation where you have only one fate to get died. Okay, there is nothing because we don't have any vaccine till now. We don't have any antiviral. Okay, even though we know dengue for more than 50 years now. Okay, so what is the conventional method that we use to diagnose dengue? It's called the direct method, which is a very primitive method where people used to do viral culture. And the modern method, nucleic acid amplification method, or the PCR method, which is always being a gold standard method. But the problem of PCR or RT-PCR, you know what? You required a very trained manpower. You required a sophisticated machine. You required some time to get the result. So you can understand its wide accessibility to a very low resource area is limited. Okay, even though this is the most accurate one. On the contrary, the indirect method of people, the daily basis people use, that is the ELISA based method, which IgM or the IgG ELISA based method, and HI assay or the hemagglutinin inhibition assay. But all this method, you know, they have their own limitations. Okay, even the other people are trying for the sensor and all like electrochemical, piezoelectric, and optical. So they are also having some kind of limitation of their own. Now, what are the limitations we have right now? Now, sensitivity. Like, what are the sensitivity that we can achieve for the dengue virus infection till today? It's not more than 60%. Okay, even though we reach specificity, like accurately, how accurately we can tell what kind of dengue or whether it is dengue or Japanese encephalitis or other viral disease, that is around 99%. Specificity is 99%, but sensitivity is not that great. And so, as a result, we have around 7% to 8% false positive result that we used to get. So, in this condition, when we don't have any vaccine available, we don't have any antiviral available, and on every year during this rainy season, India at least got affected so many people, so many children and all. So there is a need to have a point of care diagnosis, like you can do it without the intervention of medical practitioner, or without the need of sophisticated machine like PCR, RT-PCR and all. And also which will be like a self-driven test. You don't require a medical professional. So if you know the per capita doctor available, so in that condition, if you think in a very rural setup area, you always get access to the medical professional. So that is not possible. So the goal is to, or the ideal detection system for the dengue should be like this. It should be free of any medical practitioner. It should be non-invasive. It should be self-driven, and it can overcome the sensitivity issue and also false positive issue. Okay, so with that motto, with that background, we have introduced one way or one pattern. Yes,
Can I request that the presentation be on full screen?
it is
Like,
full screen. It is Mac.
sir,
So
actually on top one of the options would be present, and
hmm,
if you
that's fine now. That's fine.
can you go to the option of slideshow?
No, but
Like
there the sound.
after animation, there is slideshow.
That's fine. That's okay now.
No, actually, sir, this is not the full screen. There is an option called slideshow on the orange bar that you have. If you click there, there is going to be an option for present, and if you present that, the entire screen will become the presentation.
No, where you exactly want me? Because orange bar is not showing here. No, it's
Slideshow.
a slide.
There is a word slideshow. At least
Can you
it
see?
shows orangish from the our perspective.
Sir, your cursor is on reviews, just beside that slideshow, sir.
Slideshow.
Slide show. This is slide show, and then this one. No,
Sir, please
current.
click on slideshow.
Yeah,
Click on it.
slide show. Then it's fine.
So if you click on slideshow, actually between animations
Hmm.
and review, there is the word slideshow. Yes, that there. And then can
Sir,
you
go
please
to
start
from the
with
start, sir, the
present
very first.
from start? On the left corner, there
Left
is this
corner, sir,
green
from
button.
start.
Yes,
No,
Or
it's
from the current
from
slide, sir. You can use
from
from
the
the
current
current
slide.
slide also.
Beside that,
yeah, but
there
Yes,
is
sir.
this is inactive.
from current slide. No,
Uh-huh.
sir, no, from current slide.
Current slide
Yeah, from start and then there is from current slide. Please
from
click that.
no. This is also
Yes,
you
sir.
see. This is inactive. Okay,
Oh, it's
this is
inactive, sir.
yeah. This is inactive. So there is some issue. This Mac had some issue. Huh? So just bear with me. What I can do. I can increase the percentage.
Okay, sir. No, if there is inactive, if it's inactive or there is some tech issue,
Yeah, yeah.
then it's extremely sorry from us that we are requesting
No,
because
no,
there's
that's fine. So is it? Can you see? Means
we
Yes,
can
sir, we
more or
can
less
see.
see. We can see.
okay, okay. So let's start. Okay. So now what we are thinking, so this is what the dengue virus genome is. Okay, so if you can see my cursor, okay, this is the dengue virus. It contains three structural genes called the CME. C called the capsid, M called the membrane, E called the envelope. Okay, these are the three genes which are making the outer surface of the virus. Okay, this is called the structural gene. Now you have a non-structural gene starting from NS1 to NS5. Okay, so they are coding different, component required for the virus to replicate, to translate, and so many and so forth. So now the what we propose is that instead of looking like whatever the current, dengue diagnostic is available is IgM or IgG based or ELISA based. So they are basically outer surface antibody. So whenever this virus enters into when a mosquito bites you, this virus will be in your body, and the our immune system will raise an antibody against the surface protein. Okay, so now if you are just based on the surface protein or looking at the surface, if you are try to recognize a person or the dengue guy, then what will be happening, so this is a one way. So your variability is one, your target is one. So looking at your target, like if I give you one example, like if I want to recognize a person, you, so I will look at your attributes, physical attribute, maybe how you look out externally, how your hair, somebody hair may be straight, somebody's hair may be curly, somebody of that sort. So we are finding a attribute or an attribute to recognize a person. Same way, whatever the available system for the dengue diagnosis we have, it is based on one attribute. What is the antibody that is generated against this particular virus surface protein? So what we propose here, okay, let's target or let's look at 2 points at a time of a virus. Like we will be looking at the surface protein, like either CM or E1, and the second, the internal protein like NS1 to NS5, anyone. So for our project or for our proposal, what we have proposed, okay, let's find the target the E protein and the NS1 protein together. You understand what I mean? If you are not, please feel free to stop me. And so we are setting two target. One is the structural protein E, another NS1, which is a non-structural protein simultaneously. So we are looking. If I'm giving a real life example like before, we are looking a person head and maybe some internal part. Head may be the like an external attribute, and an internal attribute could be something inside like its enzyme level or something like that sort. Okay, and also we make a twist here, like if I am looking the E protein with antibody or aptamer, there are small DNA molecule with a specific structure and specificity. So they have specificity towards a certain DNA. Or so here we have a E protein. So we are giving a DNA counter DNA, which is like a bait. Okay. And for NS1 protein, we are using an antibody. So you understand how we are increasing the variability. So we fix the target 2, and we are looking at the two target with two different torch, one with an aptamer, one with an antibody. So totally 2 to the power 2. So basically four variables are there. So at a time, two target, two different way. If these two give the positive result, both the positive results. So with more than 95% confidence level, you can tell this is dengue. You understand? I think I am able to reach up to you. So if you find it difficult, please let me know. I can explain it again. So we fix the target two. One is E and one is NS1. One is a structural protein and one is a non-structural protein. For the structural protein determination, we are having the aptamer system, which aptamer is binding with it, and for NS1 we are having an antibody which is binding with it. So that increase the possibility of negate the negative or the false positive result for us. Okay, so it has two basic phase to execute the project. Okay, one is in the phase one, it is in the liquid phase. So what we are proposing, we are using two lipid monomer. Okay, so I would like your little careful attention here, because the whole scheme of the work is dependent on this particular first phase. So what I told you, we are using two lipid monomer. One is called the PDA, one is called the DMPC. Okay? So these two lipid monomer, as you know, lipids are as you know, lipids are molecule which is having the hydrogen and the hydrogen bond, right? So plus lipid macromolecule. So as so if you just make these two or with lipid in a polar solvent like water and all, so they will try to hide themselves because the hydrophobic nature. They will try to hide from the water, and so what they will make a circular mycelial structure. So what I told you, so use two monomeric lipid PTA and DMPC. Okay, put it in appropriate solution or polar solvent, so that it will make a lipid circle or the micelle. Now this micelle, we are functionalize the surface of the micelle so that we can add in one. We are adding the aptamer, DNA aptamer. I told you, if you can remember, just go back. What I told you, the dengue virus E protein, we are using one aptamer for it. against it. So we are making one lipid molecule with one aptamer attached to it, and same another micelle we are adding the antibody against the NS1 protein. So don't get confused. You understand? So we have two bait, we have two target E and NS1. We have two bait. One is the DNA aptamer, one is the antibody for the NS1. So in the two lipid micelle, we are adding one with aptamer and one with the antibody. Okay, and after tagging this functionalized surface, we are tagging antibody in one micelle and we are tagging aptamer in one micelle, and the rest of the portion, okay, where the untagged portion is there on top of the micelle, we are blocking it with the BSA, okay, bovine serum albumin. So you will be having this kind of vesicle with having aptamer attached with it, and rest of the blank space will be occupied by the BSA. In the other case, there will be a micelle with the antibody against the NS1 protein, and the rest of the portion will be occupied with the BSA. Now, the chemistry between this PDA and DMPC monomer and polymerization is that after monomer attached with each other, if you expose to UV light, the cross-linking will allow this particular molecule into a blue color compound, liquid compound. Okay, so when this is a stoichiochemistry, so the monomeric ratio is such a way that if you expose to UV, they will cross link and they will be blue in color. So this blue liposome, along with the aptamer or along with the antibody, will be our material for the diagnosis part. So now, if in this blue color compound, so think about this or that. So the aptamer, if a virus is coming, suppose you are giving a drop of blood, what will happen if the virus is there? So virus E protein will go and bind to the aptamer, okay? And in another case, if it is replicating, transcribing, and translating, so the NS1 protein will be there. This will be go and bind to the antibody which is attached to the one of the surface of the liposome. Now, as a result of both these things, what will happen? There will be again depolymerization of the DMPC and PDA vesicle, and as a result of what, there will be chromogenic shift or transition. Okay, so the absorption minimum, maybe I will be showing some of the data where you will be understanding the absorption maximum for this blue color compound and the absorption maximum of this purple color compound, which is after tagging with E protein or the NS1 antibody, it will be turning into little purple. There is a clear shift of their absorption maximum. So there is a chromogenic shift. So in your naked eye, what you can see is the blue color compound will be turning into purple color compound, and that is a positive test or result of having dengue virus. Okay, so this is the scheme. If you have any difficulty, please raise your voice. I can go back and explain it again. And I'll just go on to show you some of the data that we got, which is very interesting. And we are close to the completion of the first phase. And later on, I'll be explaining the second phase, what we are going to do, and ultimately how it will be look like a point of care diagnostic kit. So is there any doubt until now? Please say yes or no. Guys, you can listen to me. Hello.
Yes, sir, you're audible, and
Yes, sir,
Are you
you're
fine?
audible.
yes, sir,
Yeah.
you're
So
audible,
can
and
I
we understood
proceed, or
you.
you have any doubt till now?
So please proceed.
Okay. Thank you. So now I am quickly going to. So what was the objective? So objective was selection and modification of aptamer and antibody against dengue viral protein. So first you have to select which aptamer and which antibody we are going to use. Okay. Second, preparation and characterization of the chromosomic liposome vesicles. So as I told you, PDA and DMPC we are attaching together to monomeric unit to make a polymeric unit. So we have to characterize that. Okay, characterize in terms of what is their size, what is their charge, and what is the distribution of the charge and all. So that you have to measure. And third, chemical cross-linking of the liposome vesicle with aptamer and antibody. As I told you, so two separate liposome, one will be attached with the aptamer and another will be attached with an antibody. So that now how we are going to cross-link. So whether you will be aminated, whether thiolated, or so many modification is there, and what is your chemistry, so that you have to fix. Accordingly, you have to make the surface of the liposome, okay, and you have to modify the aptamer and antibody, okay. Like in our case, we have aminated the aptamer, and we have an already amine group is there in case of antibody. So this is amide bond that we are going to make in our case, okay. I'll be going a little from the down. Okay, now the fourth thing or the final thing, testing and optimization of the liposome sensor for detection of the dengue virus. Now, after you prepared the liposome along with the aptamer or the antibody, you have to test it whether in a real-time situation when you have a infected blood, and if you are giving a drop of blood containing a lot of viruses and viral protein and all, whether it is able to turn the blue color liposome vesicle into a purple compound or not. Okay, so that was the fourth objective. Now I am not going to the methodology. Okay, so like for example, for selection of aptamer, we are using an aptamer which is already reported, okay, against dengue virus E protein, and we are using anti-NS1 antibody. Now for preparation cross-linking, we are using a protocol given by 2005 Zhang et al. And to characterize, we are using UV visible spectroscopy, scanning electron microscopy, zeta potential analyzer, and DLS or the dynamic light scattering. Okay, and finally to test, we are doing some ELISA kind of strip. Okay, that I will be showing. Okay, now to show you some of the data. So after I have just jotted down. I don't want to go in detail. So in the first picture, you can see when we do monomer DNPC and PDA, basically we are attaching together, mixing together, and then we are allowing it to evaporate. In a what we call rotary evaporator. So in the rotary evaporator, you will be getting after mixing them in water and some other solvent, and when you allow it to evaporate, it will be making a crystallized product like this. So those are basically the polymeric lipid molecule. Now, if you take this molecule and put it in some appropriate solution or appropriate buffer and give some UV light, what you can say, as I told you, it will be turning into blue color compound. Okay, so you can see the blue color compound here, and where the absorption will be irradiated at the wavelength of around 250 or 260 nanometer. So we are not providing harsh UV because thinking that if we need to use this as a point of care diagnosis, so we may not be able to get a sophisticated UV lamp there. So we try to reduce down the irradiation wavelength, so around 250 or so. Okay, now in the three we have shown this and this, we have shown the two liposomes separately. So this is like a aptamer conjugated liposome. This is the before UV radiation and this is the after UV radiation. You can see the clear from colorless solution to blue color solution. Same way in case of antibody also, you can see this is the vesicle with antibody, and after UV radiation you can be seeing a blue color compound. So you have two separate tube. One is having this with the aptamer, one is having this with an antibody. Okay. Now next, we have asked the question, okay, if you are using the respective antagonist, like for aptamer, what I told you, E protein is the bait, and for antibody, what I told you, it will be the NS1 protein. So we use in this blue color compound UV eradicated liposome vesicle with aptamer the E protein. Okay, and we wait in room temperature for like 5 to 7 minutes, and you can see there is a clear color change from blue to purple, and same result we can get it here. Same way, if you just keep on increasing, this is a titration curve, so this is a BSA control, this is a BBS control, and this is a protein with the increasing concentration 20, 50 and 100. This is for the aptamer, so E protein has been added in a concentration of 20 nanogram per ml, 50 nanogram per ml and 100 nanogram per ml, and you can see there is no color after 10 minutes or so in the BSA tube, because BSA is not going to bind with the aptamer, and PBS obviously not. Then as you are increasing the concentration of the bait protein like E protein, your color is getting changed. Okay, and same thing happened when you are using the NS1 protein, like when you have a vesicle with antibody against NS1, and if you are adding the NS1 protein, you can see it is giving as a 100 nanogram, or color is gradually changing depending on the concentration of the weight we have in the egg. You understand? So now, after getting this thrilling result, we try to measure quantitatively what is the change or colorimetry response. Like BSA, we showed you what is the color change we can see in different level. What is the color change, color intensity getting changed, both in case of NS1 protein and E protein. And when we do the titration, I show you there is a clear shift, so you can see my cursor. So this is the absorption maxima when it is blue colored, and when we are using different concentration of the bait protein, it is clearly got shifted. So you can see the shift range from here to here. So this is a clear shift of the change. So that makes the color from blue to purple color, and it is so true for both NS1 protein and for E protein too, in case of both the bait. Okay, so this clearly tells that there is a clear transition from one maximum wavelength to another maximum wavelength, or from one color to the another color in a naked eye. You can see that, and that can be a good platform to integrate it for developing a point of care diagnostic. Okay, so I'm not going detail of the this thing, because we have even tested in the spike blood sample. Before we actually go and test to the patient sample, we in between we tested, we take the normal blood. Okay, in the normal blood we dope different concentration of the E protein and different concentration of the NS1 protein. Okay, and thinking that that will be the real time situation when a person come up with a infected sample or infected virus, with infected with the dengue virus. So the same kind of result we have got in case of BSA. Okay, FBS, BSA, PBS, there is no color change. While okay, we spike with different concentration of the NS1 and E protein, the chromogenic transition is there from blue to purple, which is normally visible and which is also depicted with the colorimetric response or the shear response data. Okay, so this is kind of give us a much more confidence that this platform is working, and this will work in future in real time situations. Okay, so this is the conclusion. Specific S15 aptamer against D and V E protein and NS1 antibody covalently attached to the functionalized PDA surface. Conjugated PDA vesicles are tested with the purified D and V E protein and NS1 protein for the detection of dengue virus, and chromogenic transition from blue to red was observed. UV visible spectroscopy, same zeta potential, and dynamic light scattering were performed in order to characterize conjugated PDA vesicle. Okay, and finally we use the spike bar sample to test the platform, and it's normally working. So that ends up the first part of the project, which we have successfully completed, and recently we have started working with the second phase. So in the second phase, so far what you have understood that in the liquid phase we are able to separately identify two dengue viral protein. One is E protein and one is NS1 protein. But think this is not a very viable option till now. So you have two different liquid platform, and you have to test separately to test the two different targets simultaneously. But ideally, a point of care diagnosis will not work like that. You have to do the experiment in a single strip or single platform, and you have to compare the result based on the single experiment. You cannot do it separately, so there is a chance of false positive result. Like one may be giving you positive, and another may be giving you negative. So in the second phase, what we are proposing is conversion of the liquid phase, that chemistry or liquid phase micelle and antibody and aptamer thing into a solid phase model. Okay, and ultimately to make a point of care strip based on the it's called the lateral flow assay. Okay, which is very common nowadays. So what we are proposing here, we have made two liposome vesicle, right? One with the aptamer and one with the antibody. Now in this aptamer and antibody liposome vesicle, we try to emulsify with a very robust protein. And if you know that some of the protein which is easily available in India, like silkworm protein, silkworm, they are the or silk producing cocoon. Okay, so they have in the cocoon they have two different protein called the seroin and the sericin, and they are very robust protein. Robust in a sense that the many of the amino acid in that particular protein is modified, not the like proline is hydroxyproline. So there are many modifications there. There are so that makes these two proteins, seroin and the sericin, a very robust protein. So what in the phase 2 we are proposing, okay, let us emulsify or mix this seroin of the sericin protein with these two separate liposome vesicles. Okay, and then make a cartridge of the ink, bio ink. Now that ink we can make a print by simple printer or so in a nitrocellulose or the appropriate filter paper with one, maybe suppose this line maybe suppose say with aptamer liposome conjugate. This line maybe say this will be a antibody liposome conjugate, and after UV it will be like blue in color. Now there will be one sample chamber where you will be giving a drop of blood. Now if the dengue viral protein is there, so it will bind with the corresponding liposome along with the aptamer or antibody, and after like incubation of room temperature, it will convert into the red shift or the purple shift. And looking at these two purple sheet positive color, you can with confidence tell that it is indeed, dengue virus, and with 95% confidence, you can tell there is no doubt or there is no chance of false positive result. So that work is underway, and hopeful that one day we can end up with having a strip with two dual target for the dengue virus, and within a span of 10 minutes, and without the need of any medical supervision, without the need of sophisticated instrument like PCR, RT-PCR, one can detect whether he is suffering with dengue or not. Okay, so that concludes my first part of the talk. So if I have not much more time, and if you have no doubt, I will quickly go to another story that we are going to do. Is that fine? Hello, guys. I have time. Can you spare me five
Yes,
more
sir, you
minutes?
have time. Yes, sir.
Okay, just yeah. So I'll just quickly go to another platform, okay, which is very associated with the recent demise of the, our beloved singer KK. Okay, he has died with the cardiovascular disease. So far, what we have understood. So, So the basic point I will just quickly go through. Basic point, So this is a typical heart ischemic condition. Okay, where you can see this beautiful heart, the outside muscle which we call the cardiomyocyte. So these muscles are very important because they pump to make the rhythm of the heart. So this muscle actually gives the systolic and diastolic pressure, and as a result of that, the heart is beating in a very rhythmic way, and the heart is coming and pumping, going out throughout the blood and all. But if you see, as these are the cells on the surface, these are the cardiomyocyte cells, and every cells require adequate oxygen in order to survive. Now, who is supplying the oxygen? The blood is supplying the oxygen, oxygenated blood. And who is supplying the oxygenated blood? By the this vein and the artery, right, which is there on top of the your heart. Okay, so now instead of sending all throughout your body the blood and all, they also need to send its own cells of the cardiomyocyte some oxygenated blood. Now because of the lifestyle, because of some many other issues, triglyceride, lipid profile, hypertension, so many and so other stuff, what happened, this particular vein, which is actually very crucial in supplying the oxygenated blood to the cardiomyocyte, they got blocked. Okay, now because of the blockage, what happened? There is a supply, loss of supply or less supply of oxygenated blood to the very extreme part of the heart muscle cells. Like this, you can see this is black in color. So that means oxygen is not able to reach up to this very distal area, and slowly those cardiomyocyte cells are getting died. As you know, if you are not providing oxygen for a long time, the cells will be having a hypoxic condition and cells will ultimately die. And these died cells is a irreversible process, means they will never come back as alive cells anymore. So gradually you are losing the cardiomyocyte cells, and you are weakening the person who are actually pumping your heart to make the rhythm and circulate the blood. So this is the situation. So now when the people get heart blockage and you have a less oxygen supply and all, this is called the heart ischemic condition. Now, in this ischemic condition, if you are not taking care, okay, or we have very little window, so within like we call it golden 6 hours. Like if you have a chest pain, you can understand that okay, there might be some problem in circulation of your blood. Now, if it is really so, then what happens? You have 6 hours. Okay, within 6 hours, you have to either go for bypass surgery or you have to have some medicine so that immediately you will not be having a complete blockage here and you will be completely losing your cardiomyocyte and ultimately there will be a stoppage of your blood or heart. Okay, so there are many, so I am not going detail, I am just keeping the thing. Okay, so now think about in a situation where you have a chest pain, like real time situation where you have a chest pain. Now, chest pain may be because of many other reasons also. Like you might be having a troubled stomach, or you might be having UTI infection, you might have so many other things. Okay, even if you sleep differently or in a bad manner, then there may be muscle which will be giving you trouble, which you will be always misguided. That might be the pain that is happening in the chest or the upper respiratory portion, that might be due to the heart problem. Okay, so there is a very subtle difference between heart ischemic pain and also the pain from the other normal region. Now you cannot ignore anyone. Like if you ignore, okay, this is because of some gastric problem and all, you might be end up with losing your heart. Okay, or if you are every time if you are thinking, okay, if there is any pain in your chest or in your heart, you might be thinking there is a heart problem or you will be having a heart attack soon. So that is also problem. So that is why the conventional technique that we have, okay, that is not sufficient to tell okay, if there is any chest pain or heart trouble is there, is it really due to heart ischemic condition or any other issue? Okay, so this is the origin of our project or the proposal. What we propose, and before I actually go, I'll just so there are many cardiac biomarker as you know, and people are using one which might be called the troponin 1 or the troponin T, topo T. Okay, so this topo T, if you can see during a heart ischemic condition, it is released in 4 to 6 hours. Okay, within 4 to 6 hours, there is a release. So if there is, this is really a heart ischemic condition, what will happen within 4 to 6 hours? The troponin T level in the serum level goes up, and that is used as a heart attack marker or diagnostic marker. Okay, but if you see that 6 hours, you have to wait until 6 hours to reach to the top of the troponin level. Like you have a ischemic condition. Now, if you wait for 6 hours, then one marker called the topo T, which level goes up. So even though this is a very good prognostic marker, but or the biomarker for heart ischemic condition, the problem is that you are already losing a very valuable time. So if somebody is having really ischemic condition and all, you are killing 6 hours to just understand whether topo T level is going up or not and whether it's a heart ischemic condition or not. Okay, so there is a need to explore some of the other biomarker. Now, if you can see HFABP, there is another the one that I am just showing you or highlighting you. This is called the HFABP. It is also known as hard type fatty acid binding protein. So that level goes up within 1 to 3 hours. You can see here. So now we thought, okay, instead of using Topo-T, if you are using HFABP, and if you can measure, if you have a technology that can measure the Topo-T level within a very short span of time, then you can tell okay, whether the chest pain that you are having, this is because of the cardiac problem or this is because of something else which you can ignore by just simply taking a some gastric tablet like pandy or something you can take and you can get rid of the problem. So from there we thought, okay, let's target HFABP in order to find a better prognostic marker for the heart ischemic condition or the CVD patient. Okay, so this is the background. Now I will not go detail. I will just show you the methodology that you are taking. So what we are proposing here is that, okay, as FABP is having several other isotype, like you can see the H is written, so it is called the hard type fatty acid binding protein. Okay, so there are several other like kidney type fatty acid binding protein. There are so there are many isotype of the FABP. So if you are using antibody against it to detect, then you will be having a cross reactivity. So instead of doing that, what we are proposing, okay, let us have find a small peptide and to screen a small peptide against this particular protein we applied a system called the phage display library. So as you know, phage display library, phage, bacteriophage. Phage is the virus which can kill the bacteria. Okay, and we have understood the molecular detail of the phage biology, so people can manipulate the phage and all. So in the phage we are introducing a random nucleotide in their head in before their pili gene. Pili is the leg of the phage. Okay, so in that case, what will happen when this random peptide in their head will be transcribed and translate? So this random nucleotide will be making a random peptide, corresponding peptide, and that will be displayed or that will be hanging. Okay, that is why it is called the phage display library. So what we are doing, so we express, we clone the gene, okay, in a suitable vector, FABP gene, HFABP gene. Then we express the protein. You can see this is the protein, 14 kilodalton protein. You can see this is uninduced and you can see the recombinant protein. Okay, we have purified this protein and then we use this protein to find out a small peptide, 7 to 12 amino acid long peptide against this particular protein. Okay, and we use the phage display library to tell you in brief. We have to code the target protein. So in our case, it is the FABP protein that we have produced in the before this protein that we have already recombinantly produced. So we code this protein in that tube, in that plate, and then dumped the phage DNA library, which will be displaying one separate peptide. Okay, so this will contain around randomly 10 to the power 7 to 10 to the power 12 different peptide. Okay, different peptide. So you can now whichever peptide having a propensity of binding with this MDGI protein or FAP protein, so they will be staying here after washing. Rest will be go. Okay, and this you have to repeat for several times. This is called the biopanning. So ultimately, you will be end up with one or the two phage which will be attaching, which will be having a true binding efficiency. Now we will have to isolate the phage, isolate the DNA from the phage, and see which one is the random nucleotide incorporated in the phage head. And from there, you have to convert it into the corresponding amino acid, and that will be your random peptide against this particular protein. Okay, so this will be the A. So you can see those are the blue color thing is the okay, they are the fudge which will be having propensity towards the. Okay, so we will be doing first round, second round, third round of the biopanning. Means whatever in the first round fudge we will be getting, we will be giving in a fresh FABP containing plate. Again, whatever fudge will be binding, rest one will be washed off. In that way, we will be repeating the panning procedure, so that you will be end up with the real true binder or the fudge displaying the peptide which will be having a real propensity or the affinity towards the APVP protein. So in that way, so you can see the number of fudges going down. In the initially this will be this much different fudge displaying different peptide. In the first round you will be having very less. Now you are increasing this particular elute, hello, and okay, uh, okay. So after getting the final round of the fudge, we are isolating the fudge DNA, then go for sequencing, and you can see this highlighted yellow one is the non-fudge DNA. Means this is the nucleotide that has been randomly incorporated to the fudge head, and if you convert them, it will be this peptide: pyridine, proline, histidine, alanine, histidine, proline, arginine, glycine, tryptophan, and glutamic acid. So these will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. This 12 nucleotide amino acid containing peptide which we have screened is the one which will be bind to the APV protein. Now this work is going on. We have also tested with the simple ELISA whether this peptide can be used to detect the level of the FABP or not. So ultimate goal is to whether this peptide can be used to in a real-time situation measure what is the level of the FABP protein. Okay, of a person come up with a chest pain. Now, if it is beyond the normal level, you can tell that this increase in the FABP level is because of the heart ischemic condition, and you have to immediately rush to do ECG, echo, or some other respiratory, ailment in order to save your life. Okay, so this is in brief. I wanted to put forward in front of you. This is how we apply our, textbook knowledge to handle some of the real-time situation that you came across. Okay. Now, without taking much of a time, I will conclude here. If you have any question, I will love to have those. Okay. Thank you. Hello.
Hello, Shubhankar.
Suchita, come up.
Good morning, Shubhankar.
Hmm.
Am I audible?
Yeah, you can highly audible. Go ahead.
Okay, students, do you have any queries?
Feel free if you have anything.
Does anyone have any query? I don't think so, ma'am.
Okay. Uh, Shubhankar, on behalf of Department of Biotechnology, Haldia Institute of Technology, I want to thank you wholeheartedly for accepting our invitation and finding time from your busy schedule for our students and sharing your work experiences. I think they have learned how much thinking and research works are there behind the development of diagnostic tools. Those are available in the market. Thank you again. And sorry for the inconveniences during the starting phase.
No, that's fine. I was a little busy. I have to go back today. So anyway, it's nice interacting with you all. I'll be happy in future if you think. Okay, there is
I
much.
will really contact with you.
Yeah. So thank you, Suchita. Thank you. Bye. I'm signing out.
Okay.
Okay.
Thank you.
Thank you, sir.